Okay, let's go for it. Okay, hi. Well, good evening, everybody. It's a great pleasure to welcome you all. As the president of the West Sussex South Branch, the Mnda, it's my great pleasure to start the meeting off. This is our 36th AGM. We'll be welcoming Nick Cole, the head of research at Mnda, later in the evening after we concluded the business part. So I'd just like to hand over to Dean to talk about the sort of technology, how we vote and how we can mute and unmute ourselves. Dean.
Sure. Okay. So in a second, I'll put everybody on mute. In fact, I'll do that now. And if you want to do the voting, if you look at the bottom right-hand side of your screen, or actually just the bottom of the screen, it says reactions there. You see the reactions button, and if you click the reactions, you can do the thumbs up if we're going to vote. So just do the thumbs up for sort of doing the vote, and if you need to sort of, you can always raise a hand if you need to ask a question as well when you're on the mute button there as well. Yeah.
Yeah. Okay.
Everyone good with that? So I'll put everybody on mute now, and then we can allow everyone to carry on. So I'm going to mute everybody.
Yeah.
There we go. And I'll just unmute Robin. Sorry, Robert, I've muted you as well.
Unmute.
There we go.
Can you hear me?
Yes. Fine.
Okay, fine. I don't think we have any apologies, Nikki. Is that right? Anyway, as far as I'm aware, we don't have any apologies. So moving on to the adoption of the minutes of the last meeting, these have been circulated, and if I could just have a vote that people are happy for me to sign the minutes of the last meeting. So if you could press your thumbs up. I don't see any thumbs down, so I will sign those as a true record of our meeting last year. Thank you very much. And I'd now like to ask Maureen to give her report, her last report. Maureen. We just need to unmute Maureen.
We planned to have the AGM face to face, and there was a lot of discussion. We had the venue booked. We were hoping to have food and drink, but we realised that actually the current situation didn't really seem to be quite right. So we decided to hold another AGM. Hopefully next year the AGM will be face to face. And I know we're all desperate to meet face to face. We took advice from the Mnda Association, who agreed with us and thought that it was actually the right decision, because people still feel very vulnerable. And they have relatives who feel vulnerable. So I think probably we took the right decision. This AGM will be my third, but my last as the chair. I don't know where the time goes. 3 years just seems crazy, really, and COVID has just made it even more bizarre. We've dealt with Zoom. We seem to have done very well, and it sort of suited us. And I think we've all learned a little bit about technology. Thank you, Dean. It's been an honour, really, to do this. I miss Sandy, and it was lovely for her to ask me to do it, but it has been a real honour, and a very, very rewarding experience. I've learned so much in those 3 years. It's been a very steep learning curve, as you all know. I've had the support of some amazingly dedicated people throughout the whole of these 3 years. I couldn't have done it without that. And I now realise really how amazing the work that is done by the branch in supporting those people with and families affected by MND is. I never understood, and I don't think we tell people enough about what we do. So many thanks to our president, Robert, Helen, you've both been fantastic throughout the whole thing. The committee, Alistair, Nikki, Sue Reeves and Judith, thank you all very, very much. You've all been fantastic. Pippa's done our newsletters for us, which has been, it's new. Again, for her, I think a really big learning curve, but I think it's been successful. Dean, Sue, Francis, Ros, Kate, you are really supportive committee members and fundraisers, so thank you so much. Without you guys, I definitely couldn't have done this. You couldn't do this without a strong committee. I'd also like to thank all of our dedicated AVs and Sue for organising our AVs. What a sterling job you do. I am in awe of you all. You're kind, caring, and you go above and beyond in helping and supporting all of our families. You are incredible. And for me, this is what the branch is about. Without you, we almost don't have a branch. Thank you to all of the AVs. You've been incredible. A special thank you to our treasurer, Alistair, who's done a fantastic job for the branch, keeping track of all our funds and keeping us fully aware of how and where the funds are allocated. He's guided me through with good advice, sage words, and the occasional telling off, which I probably needed. The branch has continued to provide support to all of our people, and it includes our magazine, the Christmas branch meeting with music, memory wall and quiz, run by Dean and Nikki, fantastic. We had a branch meeting in February, which we weren't sure if it would go down well, but it did, focusing on care and support with great talks from the AVs. They need to do more. They need to tell us more about what they do. Our accredited art therapist, Adam Levine, continues with his groups and his one-to-one sessions, hopefully for quite a while. Zoom is definitely, for him, an absolute bonus. Sue Kruger's afternoon teas, and the meetings are via Zoom with specialist speakers. And I think Sue is hoping eventually to get back to maybe going to garden centres or a coffee shop to meet. And Ros, who can't be with us tonight, had the first of our new carers support groups, helped by Sue Reeves. It's on Zoom at the moment. I think the second one's coming up soon, isn't it, Sue? I'm sure those will go from strength to strength. So we offer all sorts of amazing support, I think. It was also fantastic this year to hear that Lee Millard, who's known to many of you for his 1 to 300 blog, along with David Setters from East Surrey branch, were successful in requesting for government funding for targeted MND research so that scientists can accelerate promising progress towards meaningful treatments. And it was a fantastic talk from both of those two about how they got that and didn't expect to get it. I think, is it 5 million? So 5 million, that's an incredible amount. So fantastic for them. Branch fundraising has been really successful over the last 12 months. I think we've done very well through COVID. We continue to appreciate all of our members and their families' enthusiasm and efforts to raise money for the branch. We couldn't do it without you. And this enables us to help and support people with MND in our area in so many different ways that I never imagined. There are so many people we'd like to thank for supporting us throughout. So this year, Ham Manor Golf Club Charity Day, Joe, Parry Evans and Beth with their amazing fundraising. They were incredible. Ros and Kim Short's Open Garden, Worthing Running Club, Steve Holmes, again, absolutely amazing. All those supporting the inter-branch walk with East Sussex and North Sussex, which I think is happening again this year. It was very successful, even though it was the first time it was run. Dean and Sue for Santa's on the Seafront, fantastic. I think that's going to run again. The John Selden Golf Society, the Body Shop, who have just given us a vast amount of money, and it has been a long time coming, but again, they've had problems in terms of trying to raise funds. All those involved with the concert at St Mary's Parish Church, thank you very much, amazing. Anne Duke fundraising for Norma. Travis Swirl and his game-a-thon, he was fantastic, a young boy who did a 10-hour game-a-thon. We've also got some really great events coming up, and I'd just like to say that we have a summer fun day and barbecue on Saturday the 27th. And from what I understand, it's going to be a bumper day. It really sounds incredible. It's at Westbourne House in Chichester on the 27th of August. It has a really exciting list of stalls, classic cars, vintage fire engines, tombolas, a dog show, and masses for the children to get involved with. It would be great to have anybody that is willing to help and support get in touch with Judy, our Vice Chairman, who has spent hours pulling all of this together, and it sounds really exciting. As I say, we continue to appreciate all of our members and their families' enthusiasm. Without you guys, again, the branch doesn't run. A branch is run by a number and a group of different people. And all I'd like to say is thank you very much indeed. It's been an absolute joy. I've really enjoyed it, but as I say, a huge learning curve. So thank you all very, very much.
Can you hear me? Okay. I would like to thank you, Maureen, because you had a very difficult sort of brief. Really, you were sort of inveigled into it by Sandy, and then very tragically we lost Sandy and Lynn Crompton, two real linchpins of the branch, and then COVID. I mean, it's been a really difficult time, but throughout that you performed brilliantly. So thank you on behalf not only of everybody here, but on all the people through you that have been helped and reached with MND. So thank you very much indeed. So if we can go on now to the Treasury Report, which has been circulated, but Alistair, would you like to take us through it? Thank you.
Certainly, thank you. Welcome everybody. Thank you for your kind words, Maureen. What I'm going to do first of all is just give you a quick look at this, which you should have copies of. This is the page out of our annual report, which shows our income this year compared with last year, and our expenditure this year compared with last year. You'll notice a lot more expenditure this year, and that is because we sent money to the Chief Executive's appeal this year, which we didn't the previous year. So that having been said, let me guide you through something in a little more detail. So with the financial report for the year in front of you, here is the year at a glance, as it were, and it's income, expenditure, transfers to national office, and as a result of that, you will easily see that the branch expenditure plus the transfers well exceeded income.
Alistair, sorry, we can only see the front cover.
You should. Oh, that's a bit difficult. Let's try again. Okay, wait a minute. I think I can guess at that. I think I need to go into it before, into the slideshow first. That's what it is, I guess. So What I am having difficulty, Dean, is getting into the slideshow and then back to the
If you just reduce your navigation bar on the left and then just manually go through slide by slide rather than the slideshow, you can just sort of use the navigation to jump through. That might help you if it's not working.
That's back. Let's try this now. So you should be back at the cover.
Correct.
And if I then do that.
Yeah, that's fine. You're right. You're okay now.
That's it. That's what it was, Dean. I hadn't started the slideshow before going into it. Obviously, you can't do that. Anyway, sorry, what I was saying was that you will see from this, expenditure plus transfers to NO well exceeds income, which means that we have decreased our reserves quite considerably. But our reserves are very high, so I don't have any concerns in there. Income, this is the detail of the income, and you will see that this year a great deal of it came from our own fundraising activities. There were some donations, some in memoriam donations, no legacies, a small amount of corporate donations, and the rest was either gift aid reclaimed via MNDA or interest payments.
That's fantastic, considering the circumstances.
Yep, we'll come back to that in a minute. Coming on to expenditure, we try very hard to ensure that all our expenditure is for care. And you can see that almost all of it is. There is a small amount for information and education that covers things like the newsletter. There is a small amount on purchases for resale, a small amount on fundraising and publicity. And unfortunately, we do need to spend a little bit on administration. But 90% of our expenditure was on care for the people who we support. This is comparisons over the years since I became treasurer. And you can see that this year, our income was down very slightly from last year, but the branch expenditure was, as such, wasn't that high. Transfers to FCH were higher, and therefore there was a big change to the restricted funds. So I think if you look across the years, our performance is fairly good. This is the one that's a bit of a classic. Our fundraising over the last 12 years, and you can see that last year, our fundraising is the highest it's been in the time that I have been treasurer.
Brilliant.
Which is quite startling. We have virtually no sales because sales almost entirely come at Christmas, and we didn't really do that last year. So almost all of our fundraising was activities, and as I said, the highest in my time as treasurer. Transfers to national office. The biggest of those was to the Chief Executive's appeal. We gave the Chief Executive's appeal around about £20,000 aimed at research, and we were able to define what research that went into. If anybody's interested, ask me. Then we had the annual transfer, which we have to do. That represents half of the funds, non-restricted, which we held at the end of the year. That is on the 31st of December. So this year that amounted to around £19,000. and we asked for most of that, we asked for that to go to care and research. That's really it. So if you need to ask any questions, please feel free to do so, or alternatively, if something occurs to you afterwards, please email me at that address. I'd be very happy to add to what I've been saying tonight. Thank you very much. If there are no questions.
Thank you. Am I audible? Thank you very much, Alistair. And we do need to, it's amazing, the fundraising, that is absolutely brilliant, considering COVID and everything else. So that's a tribute to everybody, and particularly like to stress that. We do need to
some of them.
We do need to formally adopt the accounts. So could I have someone just to propose that the accounts are adopted?
I'll propose that the accounts are adopted, Robert.
Thank you very much. And someone to second that? Just put it. Thank you very much, Sue.
Yeah.
So if we could just have a vote that people are happy to adopt the accounts. Anybody unhappy with any questions for? Okay, so thank you very much, Alice. We've adopted the accounts, and thank you again for all your work over the year. We'll now proceed to the election of the branch officers and committees. So everybody essentially stands down. So I'm the only person who has a post at the moment, but that will change, hopefully. So for chairman to take over from Maureen, the nominee is Barbara Albon. She's been proposed by Alistair and by Nikki. So with no other people being nominated, if I could have your votes for approving that nominee. Any against? Well done, Barbara. I congratulate you on being adopted, and really very, very welcome addition, and having a great start with the St Mary's concert already. Thank you very much. Now, vice chairman, the nominee is Judith Allen, who's been proposed by Maureen O'Neill and by Alistair Sharp. The only nominees. So again, if I could have your votes in favour of Judith, and none against. So Judith, congratulations on being reappointed as vice chair. Secretary, we have Nikki, who's been proposed by Dean and seconded by Sue Kruger. Again, no other nominees. So if I could have your votes there. Thank you very much. I don't see any wrong. So congratulations, Nikki, on being reappointed as secretary. And treasurer, nominee is Alistair, who's been proposed by Nikki and seconded by Judith Allen. No other nominees, no one anxious to take over as a tough job of treasurer. So if I could have your votes there. Thank you very much. I don't see any dissension. So congratulations, Alistair, on, is it now over 10 to 11 years or? Sorry, being muted, but it's certainly approaching.
As I said, it has been 11 years.
Whoa, very good, very, very impressive. Now, the committee, we usually elect the committee en bloc. So I'll just read out the committee members, which are Maureen, who's now standing on as chair, but is on the committee, Kate Taylor, Sue Kruger, Francis Ramsden, Dean Donaldson, Pippa O'Dwyer, and Ros Davis. And by tradition, all the association visitors are co-opted onto the committee. So I'll just read those out. That's Sue Reeves, Angela Burgess, Anne Coleman, Prue Tyrrell, Patty Shaw, and Valerie Sharp. And if I could add my thanks to Maureen, to the fantastic work that the AVs do. I can know from my times as a neurologist that it really is a fantastic support that they offer, and I would really like to commend their work. So if I could have I'll need a proposer for those committee members. So I think.
Well, I can propose.
Yes, you can do that. And seconded.
I can
Thank you very much, Dean. So if I could have a show of approval for election of all the committee members en bloc.
And then we're going to adopt some people, the new people.
Thank you very much. And I gather there are some new committee members. Adoption. So Barbara, would you like to just speak to that?
Yes, it is Claire Angles here. This is Claire,
Claire?
my friend and neighbour. And this is Frances Clinton.
Hi.
Yeah, both ladies would very much like to be involved on the
Right.
committee.
So we'll say Barbara's proposing.
So we're proposing, if I could just have a seconder to Barbara's proposal there. Thank you very much, Sue. And a reaction,
Show of hands.
a show of hands. Fantastic. So welcome aboard, ladies. Fantastic. Right. Okay. So so that concludes the election of the branch officers and committee. Now it is exactly 7:30. We would be on the agenda of the plan is to go on to Nick Cole to give up it.
Robert, there's another proposal as well, if I can.
Please, please.
Sue Caster, who wants to join the committee.
Oh, fantastic.
Oh, right, brilliant.
Okay, so if we can have a seconder for that.
I'll second it.
Nikki, thank you very much. And a reaction? Put your hand up or? Right, fantastic. So welcome aboard. This is really, really good news. Because, the bigger the team, the better. Fantastic. Thank you very much, Dee. Have we heard from Nick at all about whether he's joined us?
He's just joining now.
Okay.
Okay. If I could just say a few words while we're waiting for Nick to get on board. Thank you very much, Rob, and everyone for voting me on the committee as chair. And I follow in some very skilled and professional footsteps. And I'm honoured that Maureen asked me to follow her into the role. And again, thank her for her amazing work, the energy and enthusiasm she brought to the branch. As said before, through some extremely challenging times. I'm very much looking forward to getting stuck in, as it were. And as this is all very new to me, I welcome any advice you care to give me. I've always known that I would support and work for the MND Association one day. I didn't quite expect to slot into the role of chair quite so soon. However, as the MNDA is very close to my heart, I'm very much up for the challenge. So, cheers. And is Nick here?
Yes.
Yes.
Okay, hello Nick. And I've now great pleasure in introducing Dr. Nick Cole, who is Head of Research for the Motor Neuron Disease Association. I had the privilege of hearing him speak at the Francis Crick Institute in 2019, and I can assure you his talks are inspiring and he speaks in a language we all understand. This evening, he will be giving us an update about MND research and what is happening today in MND research. And welcome and thank you, Dr. Cole, for giving us your valuable time. And over to you.
Thank you very much. Wow, that's
Hi.
hi there. Can everyone hear me all right?
Yep.
Great, thank you. Wow, that was quite an introduction. That's something to live up to. Ultimately, if you have any questions, you can just ask me them as we go through, or at the end we'll do ask me whatever you like. That's fine. So thanks very much for the honor of being able to talk to you today. That's great. So I'm just going to give you a bit of an overview of the work of the association, which I'm sure you'll be more than familiar with a little bit of science, and then we'll kind of build up into some of the things that are happening in research at the moment. And as I say, if you have any questions, etc., and I'll try and signpost you to any information, etc., that you might like to look at later on if you have time. Now, Robert, you've got a hand up there. Is that from a previous hand up, or?
Yeah, I'll remove that.
No problem. No problem.
As you haven't said anything, it's a bit premature, I guess.
Excellent. Yes, great. Okay, so let me just see if the magic of sharing screens works. Hopefully it will. And there we probably go. Let's just try and make that full size. Okay, can you see that heart shape with the pictures?
Perfect.
Oh, that's a relief. Gosh, you never know. Sometimes it does, sometimes it doesn't. Great. So yes, so I've worked now for the Mnda Association for just over 4 years. I'm in the research development team, so we're in charge of research communication, trying to put across the latest developments in research. And within the research development team, we have the grants team, so making sure that every penny that's donated goes to the right place in terms of research that we want to fund, among other things. And we also organise the International Symposium. One of the roles of having our information, we have a great website, we have a blog, and we have an email address and a phone number. So if you have any questions, you can always get in touch with us if you want some information. We have sign up newsletter, which can give you the monthly latest research news as well. There's a huge amount of things going on in M&D at the moment, as I'm sure many of you are aware. Too many to mention everything in this, but hopefully we can just touch on a few of those things. So we've just had a sort of redesign, as it were, with promises, which I'm sure you will have seen as well. And the research promise is that we won't rest until M&D is treatable and ultimately curable. So that's really my job and everyone else's job as well, including our community and yourselves, to try and make that happen. We've been trying to do this for some time, but obviously it's a very difficult and complex disease to understand and to find effective therapies for. But there are some good steps being made and some progress being made, and hopefully we can go on and talk about some of those things. So the ambitions of ourselves and the team is that everyone who wants to take part in research is able to. At the moment, that doesn't really happen. There are some restrictions in criteria for people to take part in research. Some of that has changed post-COVID, with people being able to take part in projects remotely. And people with MD are able to access effective treatments as quickly as possible. We increase global collaboration, so we do this through the International Symposium, which we organise every year. And last year we had 1,400 delegates representing 45 different countries, so a real melting pot of all the research and healthcare professionals involved in research. And generally, the more we can fund and the more projects that go on and the more results that come out of those projects, the greater the understanding of MND, the biology of MND, because ultimately without understanding the biology, it's very difficult to find effective treatments. It's difficult to understand what's going on in the nervous system because it's well inside our bodies. So it's a complex and difficult to study. As I say, progress is being made. Hopefully we'll touch upon that as we go forward. So we have lots of resources. We're a hub really for researchers and for general knowledge for the public as well. So we try to put out as much information to try and dispel the myths. We don't want to build false hope. There's a lot of competing interests between researchers, pharma, drug companies talking about results that may or may not be as promising as we feel. And so we really do a deep dive into that data and try and make it understandable. So lots of information on our website. If you want to ever take a look at that on the research section of our website. We have the latest news regularly, and also trials that are available. These are two of our blogs which talk about clinical trials that are ongoing at the moment in the UK and wider, and I'll mention some of those as we go forward. So we've got an M&D research blog page and also a latest news page. So what's the latest news in M&D research? And anytime something comes out, we put a little tag here on the website to keep an eye on new publications, clinical trials, etc. So we scan globally really to see the latest news and developments, and then we share that information with our researchers. So as I'm sure you know, what is motor neuron disease? It's a disease of the motor neurons, which are the electrical wires that connect our brains and spinal cords to our muscles. So there's an almost a branding issue in terms of, in the UK, we call motor neuron disease because it's an umbrella term for diseases of the motor neurons. But in America and a few other countries as well, including France, you'll have heard the term amyotrophic lateral sclerosis, or ALS, which is actually the medical term that doctors use for around about 90% of MND particular type. and there's also a few others there, including PBS, PMA, and PLS, which are really the same principle, that the electrical wires that send signals are degenerating, and but they're named in different regions because of where the first symptoms start. So motor neuron disease is the umbrella term for MND. As I've mentioned, it's the electrical wires that connect our muscles to our brain and spinal cord. So when you wish to voluntarily move, say, your little toe, you're sending a signal from your brain down your spinal cord through these motor neurons to allow that muscle to contract. And because muscle is actually a plastic tissue, so you can train muscle, you can lift weights or run a marathon, or if you have your leg in plaster, or you spent 6 months in space, as these cosmonauts here in the picture show, then muscle, if it's used, it gets larger and you can increase the number of fibers in the muscle. If it doesn't get used, then it starts to atrophy and waste away. And unfortunately, that's what happens in motor neuron disease, because the electrical signals are not getting to the muscles and the muscles are not being used, and the muscle starts to get smaller and degenerate, and eventually those voluntary muscles, breathing, swallowing, locomotion, are no longer able to form. So the question is, why does this happen? So these cells, motor neurons, are single cells, and they don't regenerate throughout our lifetimes. And this can be a single cell, could be up to 1 meter long from your spine to your little toe. And like every cell within the cell, there's a cell body, which makes up most of the cell, with a nucleus in the middle. And the nucleus is actually the control tower that sends out the signals for what the cell should make, what proteins are needed to keep the cell alive and to perform its functions. But in motor neurons, you've got this really long axon, as I said, up to 1 meter long, which goes into the terminals, which join with the muscles. So these are neuromuscular junctions, so the junction between the nerve and the muscle. And there you get a chemical signal from calcium and other things, which is made from an electrical signal. So a lot of complexity going on. And we do know that within around about 95% of people with motor neuron disease, one of the proteins in this nucleus or this cell body, which is called TDP43, the name isn't really important in this context, but that protein should be within that control tower or within that nucleus. But in motor neuron disease, the protein ends up outside the nucleus. And there's a few other proteins, only 3 others, which are also do this similar clumping outside the motor neuron in MND. So this is a potential avenue for a therapy to try and make sure that these clumping of this protein within the neuron doesn't happen, and to try and keep that protein in the right place. And this is a slide showing that process of the protein TDP, which is normally in the nucleus, starting to spread out cell in a case of motor neuron disease. And as I said, around about 95% of cases, this protein's in the wrong place. So there are some very clever targeted therapies that are being developed and are in clinical trial to try and stop this happening by actually stopping this protein from being made altogether, to make sure that it doesn't end up in the wrong place. We'll talk about that in a little bit. So it was originally thought that motor neuron disease was all about motor neurons, hence the name. But actually, we now know that there's a huge number of cells involved in maintaining the motor neuron. So around that cell body, for example, there are cells called microglia, and these are cells which are involved in stopping infection, fighting infections and any foreign bodies in the nervous system. Astrocytes, which are cells which actually nourish the neuron, provide it with nutrients, etc. And the axon has this very complicated cargo system which shuttles energy up and down the neuron, and nutrients and things that the neuron needs down at the neuromuscular junction by the muscle. And again, this very specialized structure, the neuromuscular junction. So any of these different structures plus others could be involved in motor neuron disease. So lots of projects are trying to understand this biology. So why is it that this happens in MND? And to stop it happening. We know that around 5,000 adults in the UK are affected at any one time, and that number of people will significantly increase if an effective therapy is available, not necessarily a complete prevention. But this is why pharma's greatly interested, drug companies, in M&D, because actually an effective therapy would be something that they could potentially sell continuously to prevent or slow down symptoms. So this is something that we're trying to capture that interest from pharma, because we know that that's where lots of the funding is in terms of developing effective therapies, and that drive is there to take these ideas from universities, for example, through to actual effective medicines. And that's something that we're focusing on going forward, something called translation. So translation from ideas through to medicines. So is that okay so far? Makes sense? Okay, great. So as you'll know, everybody's journey with MND is very different. So some people are able to live for a long time. Some people have a very fast rate of progression, and we don't really know why that is. Everybody is different. There are clearly some factors involved which allow people's progression to be slower than others, and also there are pauses in people's progression. So sometimes people will progress rapidly, and then their progress of the disease or symptoms will slow down. There have even been a very, very small number of reversals in the world. So this graph is actually showing that rate of progression of a whole population of people with motor neuron disease. So each line represents a person with MND, and we have the ALS-FRS revised on the left-hand side. So this is a score from 0 to 48 of measurements of progression, 48 being somebody without motor neuron disease. And there are various scores in different categories, for example, locomotion, handwriting, speech, etc., and time across the bottom. So the reason I'm showing you this is because this heterogeneity or the way that people progress very differently with their disease makes performing clinical trials quite difficult, because interpreting the data is quite complex, because that pause in progression could occur with somebody in the trial who's on the drug or on the placebo, and that would make the researchers feel that actually the drug is working when in fact it's just a natural progression. So this is something that has to be sort of taken into account as we go forward. So one of the ways to overcome this, and is done in other diseases such as cancer, for example, or even in diabetes, is to have a biomarker which allows you to get a biological readout of the rate of progression and for diagnosis. So that's another area that we're looking at the moment. We'll come to that as well in a second. So 6 people are diagnosed with MND every day. And so what are we doing about this? Well, as you're all very, very much aware, we're involved in care and quality of life improvements for people with MND, campaigning as much as we can to raise awareness of the condition and try to bring as much funding for people as possible, and research, which is where we're at and which I'm going to talk to you about. So we try to fund the very best research we can, the very best people to uncover new and effective treatments. At the moment, we have just over 80 grants being funded. So that's around about 200 individuals being given part of their salary to do research into MND. And we have a portfolio of all the projects that we fund, again, on our website. As I say, 82 different grants, too many to go into at the moment in this time. And there are various different types of grants. So we fund people, the very best people we can, PhD studentships, as you will know, and also projects and clinical research fellowships. So people who are clinicians who have 1 foot in the lab and 1 foot in the clinic, and non-clinical fellows as well. So people who are funded with their salaries to do research projects, and small grants, for example, sending off some samples to be sequenced to get some genetic information, etc. So a wide variety of different projects, different types of grants, but we try to leverage as many as possible with other funding, and around about a third of our grants are funded with other organizations, which is a really good way to get good value for money. For example, our clinical fellowships, as I talked about, where one particular person is a clinician and also performs research in a lab. This is at the Crick Institute, Ricky Patani. For every £1 that we put into that project, the Medical Research Council gives us £3.75 for his fellowship. So that's a good way of leveraging every £1 that goes into the tin, however that's collected through fundraising, etc. Okay, so this is quite a bit of information on this slide, but this is really a breakdown of our kind of research portfolio and how we break down the research. So we have a series of different things going on. The M&D International Symposium, as I mentioned, and an M&D register, Project Mind, an M&D collection, which is all the samples that are collected from as many people as possible living with M&D and not, for researchers to use. And then we break down the research into causes, models, developing treatments, markers of disease, and healthcare. And I'll touch on some of those in detail in a second. So if we're trying to understand the causes of motor neuron disease, why is it that these motor neurons are affected in M&D? Why is it that different people have different rates of progression? Where do people with M&D, where are they in the country, so we can direct the best care we can? Is there anything specific about what careers people may have had with M&D? So part of that answering those questions has been started by beginning the M&D register for England, Wales, and Northern Ireland. So people who are diagnosed with M&D are now automatically opted into the M&D register, so we can gather some information as to where people are. So you could perceive that there are lots of people on the south coast of England with M&D. Why is that? Is that because of some environmental issue, or is it just simply because there's an older population in some particular places? So that's given us that information. And also what causes M&D. So we know that in some cases motor neuron disease runs in families, and that's an indication that there's a possible genetic cause. So one of the projects that we're working on in a large collaboration is something called Project Mind, and this is actually sequencing the DNA, so the blueprint of 17,500 people with motor neuron disease and 2,500 people without motor neuron disease, to try and identify genetic causes of the disease and even protective genes which might be protective. So perhaps somebody with a slower rate of progression has a protective gene than somebody with a more rapid rate of progression. And this has proven to be a really successful collaboration that we've put some significant funding into. And there are now around about 44 different genetic different genes which are associated causing motor neuron disease. So we've provided around about 10% of the global total for this particular project. So as I mentioned, we know that in 10% of cases of MND, there's a family connection, and around about 90% of cases are what's known as sporadic. So the disease seems to appear for no reason, no biological reason that we can identify. It doesn't run in the family. So why does that happen, and can we understand more about that? And actually, it turns out that in sporadic cases, some of the same genetic causes that cause the disease in sporadic cases also are the same genes that cause the disease in familial cases. And this is really significant because it allows us to target what does a particular gene do? What's its role in our bodies? What pathway is it affecting? And that gives a potential target for a medicine against that particular gene or protein. So genes make proteins in our bodies, and if the protein is made wrong, then a disease can potentially begin. So this is just showing a graph of the rate of gene discovery, and there are really there are three main pathways that are associated with these particular gene discoveries, and so that's three different pathways to target with therapies and potential medicines, and some of that research is ongoing. I mean, lots of research is ongoing to try and understand this protein homeostasis, which is the balance of proteins, RNA binding proteins, and cytoskeletal proteins. So the proteins that actually make the motor neuron, gives the motor neuron its structure. And a paper that came out this week has actually looked at these sporadic cases, looking at the genetics of sporadic cases, so people without a family history, and shown that around about, in some cases, people have 18 different genetic changes which are causative of MND. So if you have that whole combination of gene changes, you may be susceptible to motor neuron disease, as opposed to people who didn't develop motor neuron disease. So the genes and these genetic studies are very powerful, and they are really giving us a handle on the biology, what it is that's causing MND. So if you think of the genetic addition, the addition of the genes are like grains of sand on a scale. So you have some environmental factors. Everybody's had different environmental exposures. We don't know what environmental exposures cause MND by any means. We've all got different lifestyle, lived different lifestyles, and been exposed to different things as well. And then genetics. So you've got these grains of sand on the scale, which may cause development of symptoms of MND. So that's causes. Models of MND. So it's very difficult to study the biology of MND inside a person because you can't actually see what's going on. So people model the disease. One of these really amazing ways of doing this is to use what's called pluripotent stem cells. And pluripotent stem cells are cells that can actually make any tissue. So these two chaps who won the Nobel Prize for this research discovered these factors called Yamanaka factors. So we can actually take a skin cell, a fibroblast skin cell from any person, and if you add these 4 different chemicals, which are just different proteins, at the right time in coordination, you can turn a skin cell into a stem cell, so a cell that's got the ability to form any tissue. And then you can perform a second reprogramming of those cells, and if you add the right factors, you can then turn it into a motor neuron. So researchers can take skin samples from people with MND and skin samples from somebody without, and actually grow those motor neurons in a dish, and then perform experiments to see why the MND motor neurons are different to the non-MND motor neurons. And this experiment here from Oxford University is an example of that, where there's a bit of green on the right-hand bottom figure, and that's actually a marker of stress, of oxidative stress in tissue. And so if you look at the motor neurons on the top right, the blue's the nucleus, the red is the axon. There's no green in that dish, and so those neurons are working effectively with no stress, no oxidative stress. But the ones in the bottom right have this green color showing that they're under considerable stress. And so by applying different compounds and manipulations to those neurons in that dish, if you can get rid of that green color, you may have an avenue towards a therapy. And this is where it's ongoing in Oxford. As I mentioned, markers of disease progression, really important. So as you'll be aware as well, diagnosis is really, really difficult in MND. It's more a process of elimination for a lot of people. It can take a huge amount of time. And so finding a marker of MND is really important to speed up diagnosis, and as I mentioned, in clinical trials, to get a good effective readout of disease progression. So one way to do this, ignore this slide, there's a lot of information on there, but actually the most recent and what looks to be the most effective biological marker for MND is something called neurofilament light chain. And this is results from a recent clinical trial of a gene therapy, which actually showed that the level of neurofilament dropped in people with MND when they were given the medicine. So in motor neuron disease, neurofilaments are like scaffolding, which actually the neuron is made from. So as the neuron degenerates and breaks down, you end up with this neurofilament, or these bits of scaffolding, in your bloodstream and your spinal fluid. And so you can measure that as a level of neurological loss. And so by being given this medicine, if the level of neurofilament light chain falls, that indicates that the drug is hitting its target, and the toxic proteins that are involved in this particular case, where the medicine is applied to, are being removed, and so the level of breakdown of nerves is starting to slow. And a project for a second set of biomarkers, which is going on at the University of Sussex. You may know Greg. He may have spoken to you already. I'm not sure. And he's looking at RNA as biomarkers, and he's found seven RNA molecules. They're particular little molecules in the bloodstream, which people with motor neuron disease tend to have higher levels than people without. And so this is another potential biomarker, and we've just funded a follow-on grant for this to try and look longitudinally in people, so over time, whether these biomarkers change as the disease progresses. We've got a research blog on this on our website, and Greg's made a really nice video to explain this project. This is a really great piece of cutting-edge, groundbreaking research for M&D, and lots of people internationally are interested in how this proceeds. Does that make sense? Are we all right for time? Do you want me to slow down, keep going?
No, we're fine.
Excellent. Okay, great. So one of the other really important approaches is healthcare. So at the moment, there are only 2 compounds that are licensed for motor neuron disease. They don't have a don't make a great deal. They're not really effective. They're not as effective as we would like. There's only 1 licensed in the UK, which is Riluzole. And healthcare, quality of life, and multidisciplinary care is something that actually extends life far more than any drugs that are available at the moment, unfortunately or fortunately, really. And so there are many anecdotal conversations, and people have been treated in many different ways all over the world to try and make improvements. So one of the important things about our healthcare projects is to try and get evidence, so evidence-based healthcare. So is maintaining body weight, does it have a positive effect? So we fund projects in care and healthcare to make sure that we can try and maintain quality of life as best as possible and get actual evidence for which particular things are the most effective. So lots and lots of different projects based on healthcare and quality of life. Again, on our website, we have a list of all of these things that are funded with lots of information and videos, etc. So that's 4 of the pillars. The final one is developing treatments. So this is a bit more of a longer explanation, but of course we're all very interested in developing therapies to slow down and stop MND completely. So one of the projects that we funded is called Myricals. So I mentioned at the beginning these cells called microglia, which are actually protective, and they prevent invaders from getting into the nervous system. And if things do get into the nervous system, then they can engulf those invading cells or toxins, etc., to get rid of them. And this is what happens in neuroinflammation. So neuroinflammation, or inflammation, really is one of those things where you have an injury and lots of cells go to that site of injury. So in motor neuron disease, neuroinflammation occurs. So there's lots of cells, and these microglia cells that go around the neurons when they're under stress. So as the neurons become under stress in the disease, these cells called microglia, instead of being protective, they actually turn onto the motor neurons themselves and start to engulf the motor neurons. So one of the things that this clinical trial is doing is using a drug called interleukin-2, which is an already approved medicine. And the good thing about using an already approved medicine is that it doesn't take as long to get it approved for another disease. This is approved in cancer, actually, interleukin-2, but at a low dose, it's been shown to increase the level of cells called Tregs. So if you increase Tregs, they're a bit like bouncers in a nightclub. So they can take these microglia cells and just say to them, hey, calm down. You need to be Luke Skywalker, not Darth Vader. Just do the right thing. And so in the earlier phase of this trial, it's been shown that if you take low levels of interleukin-2, you can increase the number of these Treg cells. So you get more bouncers in the body, and these can then have a positive effect on microglia to calm them down. And so this trial's fully recruited. This is Nigel Lee, as I'm sure many of you will know, again from Sussex Uni, who's headed this trial. Again, another international collaboration. So this is a really good way to do research, is to get as many countries, best and brightest people involved as possible. And this trial will hopefully read out towards the end of the year. So we're very hopeful that this will have some positive results. And again, another trial that we've which is now fully recruited and will read out towards the end of 2023, is a trial using something called Tutco, which is actually an antioxidant, and it's originally from the bile of bears. There's no bears involved anymore, I assure you. It's chemically made. But the reason I show you this is because there's another clinical trial that's gone on in the States for something called AMX0035, which has received a huge amount of media attention from a company called Amylyx. And that's a mixture of Tutco and phenylbutyrate. However, this trial, again, has been fully recruited and is ongoing and will read out in hopefully by the end of 2023. So this is an example of clinical trials that we have funded and are ongoing, looking at therapies, effective therapies against MND, we hope. But these are single trials. So this is where you bring in all the infrastructure and you do a single trial in a single place. You get the staff in, you train everybody up, and it's quite an expensive and it takes a long time to do and to recruit everybody. And every trial is different, with a different set of regulations and a whole new set of people on placebo in every trial. And so it's a bit like, this is the ice hotel up in the Northern Hemisphere. So every year this hotel gets built out of ice and it melts every year, and then they build it again. So it's great, it works really well. But again, it's kind of expensive, and it's a long drawn out process, and probably not the most efficient way to have a hotel, albeit that it's not a bad place to stay. So this again is coming from cancer, is this way of doing platform or what's called adaptive trials. So in this type of trial, you actually have multiple drugs being tested at the same time. And the placebo, so the group that are given the dummy drug, those people are actually shared amongst the other, can be shared, their rate of progression can be shared amongst all the other drugs at the same time. So it results in less people being on the placebo and more drugs being tested. And the reason it's called adaptive is because you can actually take drugs out of this trial if they're shown to not work and put new ones in. So it's a continuous conveyor belt. And all of that infrastructure is already in place and remains in place for the whole time. So a really good and efficient way to do drug trials. And that's happening now for MND. There are 3 different platform trials ongoing, one in the States, one that you will have heard of, which got a huge amount of media attention, called MND Smart, which is testing 2 drugs, memantine and trazodone. And lots of people have tried to get onto this trial, and it's been somewhat oversubscribed and delayed by COVID, but it's trying to get on track. And it's quite a small platform trial, which isn't really doing a lot of analysis necessarily as much as the latest one, which I'm going to tell you about, which is called the Magnet trial from Tricals, which is about to launch. But MND Smart has just been assessed on stage 1, sorry, that's shown that actually there's no really adverse effects from the drug, which is what you'd expect, because they're actually again repurposed drugs in this trial, and so it's continuing on, which is good news. So there's potentially 2 compounds there, and we are part of a network called Tricals, which is 15 different countries, and we've launched this platform trial called Magnet, which is actually testing multiple compounds. I'll just move past this gene therapy and come back to it in a second if we've got time. So Tricals is again a consortium, so lots of countries, including Canada and Australia, and it represents 46 different research centres in 15 countries. So this platform trial system is started now through Tricals, and it's called the Magnet trial, which is a multi-arm adaptive trial, and Magnet is an acronym. And it's just enrolled its first patient in the first drug, and these drugs are starting to be tested, are going to be tested in the UK starting in a few weeks. So the first one is called a Prelude trial, which is testing lithium chloride. So it was shown in a couple of trials previously that when people with MND were given lithium chloride, not everybody responded, but there was some hint of possibly a response. And the samples that were taken from people in the trial were kept. And then they were analyzed later after the trial for the genetics, what genes the people carried. And it was shown that that people who carried a gene mutation in a gene called UNC13A, UNC13A, the name isn't really important, but a particular gene change in some people with MND, their MND seemed to slow down, their progression seemed to slow down. So it was possible that lithium chloride was effective only in people with this UNC13A mutation. And so this Prelude trial is now starting in the Magnet platform, and has started in Europe, and the UK is trying to get itself up to speed with these trials starting in the next few weeks. So that's one particular trial that's ongoing, and a second one in the Magnet trial is called Lighthouse 2. This is actually an antiretroviral drug. So it's based on HIV medicine, which is antiviral, and again, you'll have antivirals being used in the fight against COVID. And this is because it's possible, and some data has shown that MND, people with MND have a reenacted retrovirus from all of our genes. Everybody has these ancient retroviruses throughout our DNA. They make up a huge proportion of our DNA. They don't actually do anything, they just sit there dormantly, but it's thought that maybe these ancestral retroviruses are being reignited in people with MND. So this is an antiretroviral therapy that's been tested in the Magnet trial. So in addition to that, as I said, the platform trial, there's this Amylyx drug, AMX0035, which is a mixture of Tutco and phenylbutyrate, an oral edaravone trial as well, the Triumec and the lithium carbonate. So these are all going to be starting in the UK in the next few months. So do you want me to stop? Have we got time? I'll just touch on some of the gene therapies. Is that all right? Yeah, okay, great. So I talked about genetics. So we know that several genes are associated with causing motor neuron disease. So, and I mentioned that genes make proteins. So these proteins that are made in people with MND, if you have a particular gene change, because genes make proteins, the genes can make the protein wrong. And if a protein's made wrong in our bodies, instead of doing what it's supposed to do, it can interfere with a particular pathway. Sometimes it doesn't do anything. Sometimes it's fine. People have lots of gene mutations all the time. Everybody carries thousands and thousands of gene changes. But sometimes a gene change can cause a toxic problem in the body. So this is the analogy that you've got all the bits there in your DNA and you put it together and you've got a fantastic chair from IKEA. But if you put all the bits together wrong, which is what happens when a protein is made incorrectly, it's not really going to perform the function you expect it to do. And this is how proteins are made. So all of our DNA, the blueprint that makes me, and you, every cell contains all of our DNA to make a whole person. But only particular genes are activated within each cell. So in that whole huge amount of DNA, the genetic code, you have a few areas which are particular genes. Now genes are photocopied. So we just look at the top section of this slide. The gene is photocopied into something called RNA. And RNA is then coded by these building blocks called amino acids. And the amino acids are joined together in a very specific order based on the coding of the RNA, which is based on the coding of the gene, to make, and then folded in a very specific way to make a protein, and the protein works. Now, in the case of SOD1, for example, which is a gene that runs in families, which causes MND, you have one particular change, a base pair, an error, or a spelling mistake in that gene. That spelling mistake is photocopied into the RNA, and then that photocopy mistake then causes the wrong amino acid or the wrong building block to be built into the protein, which means the protein doesn't fold properly and causes toxicity. So the theory with gene therapies is that you can stop making the toxic protein if you can stop making the RNA. So the RNA is made, but then if you bind something to that RNA, you can actually make it be broken down by the machinery of the cell. So you never make the toxic protein. And the three key proteins in MND, which we know are outside the nucleus, these clumpy proteins, are something called TDP43, SOD1, and FUS protein. So clinical trials are ongoing at the moment to try to target these particular transcripts using gene therapy. And as I showed you in the neurofilament example, this is for the SOD1 gene. When that gene therapy is given to people, the neurofilament level actually drops, which indicates that possibly the therapy is reaching its target and lowering the amount of toxic protein, which you would hope would mean that the MND is stopped because that protein is never made. Now, unfortunately, in this clinical trial, the people in the trial, their ALS-FRS, or rate of progression, their progression score, was no different to people who were given the placebo, the dummy drug. But it's thought that the reason for this is because by the time people have been diagnosed, that it's possible that so many neurons have been lost that it's very difficult to recover from that with the therapy at such a late stage. So the analogy being that perhaps if you went into a building that was burning down, if you went in very late, it's very difficult to bring it back how it was. But if you can stop this early enough, before the burning happens and everything is lost, then the therapy will work. And so the next phase of this trial is actually taking people who are carrying the gene, the SOD1 gene, which may cause the disease, before they have any symptoms, and measuring their neurofilament light chain level. So before, you may carry a gene for MND and have a normal neurofilament light chain level. And then when symptoms are about to happen, or the disease is about to happen, your neurofilament chain level goes up significantly without any symptoms. So as soon as they find that in people that are in the trial, then they give them the medicine. So the medicine's been given to them way before they develop any symptoms, and hopefully that will head off the toxic protein before it starts to be made and causes any loss. So that's something that's to watch out for, that we're excited about, and hopefully will prove positive. Another gene, FUS. So this is a lady called JC. She's an identical twin. Her sister died of MND early on with this FUS gene, and again, you can see in the left-hand side of this image at the top left, people in a control without the FUS mutation, the FUS gene, have none of this aggregate protein, which is the stain brown in these cells. And then with this change, this gene change in FUS, you see this brown stain. And so this medicine was made specifically for this lady, and unfortunately, she died not from the medicine, but because she had very severe MND. But on the right-hand side figure, this is actually samples from her kindly donated, and this aggregate protein disappeared from her tissue, wasn't there anymore after taking the therapy. So it shows that, again, the therapy is hitting its target, and it's hoped that if that can be given to people early enough, then it will stop them developing symptoms in the first place.
DNA.
So there's some pretty amazing people doing some very, very clever things and working really hard towards MND. I'm just finishing now, don't worry. So we've taken a good hard look at what's going on in the global research field. You know, why is it that we're not getting these therapies through? What's happening? There are lots of good ideas. There are many drug trials going on, which is great, but we need more and more things to be tested. And lots of ideas and papers are being produced, but really there's a bit of a gap. So universities coming up with great ideas, producing all this data, all this information about MND, but we're never quite getting it towards medicine. So there's this gap in translation, as it were. So only a few things are getting taken towards clinical trial. So we want to make a significant difference to that, and that's the basis of our most recent approach. Again, collaborative approach using LifeArc, My Name's Doddy Foundation, MND Scotland, the MRC, the National Institute for Health Research, And we're trying to bridge that gap between ideas and taking things into pharma. So we started off with various effective grants, a £1.5 million collaborative funding call, which we've had all the applications for, and they've all been judged, peer reviewed, to try to take more and more compounds through to potential therapies. And also you'll be very familiar, especially if you saw Breakfast TV this morning, about the 50 million over 5 years that the government has promised us to build this virtual research institute. So this is five universities, big M&D research centres in universities in the UK, and our 22 care centres and charities all together. So we're hopeful that this is really going to kickstart more and more research and bring pharma to the party to make sure that they back and really concentrate on finding effective treatments in M&D. So as I said, if you need any information, you have any questions, we've got lots of sources for that, but also you can contact us. I realize that was a very long talk. Thanks for bearing with me.
Thank you very much.
If you have any questions, please fire away if there's time.
Thank you very much indeed, Nick. That is brilliant. Yeah, has anyone got any questions?
Yeah, hi, I've got a question. I was interested in what you said about this recruitment of people before they've got symptoms. How would you get, because a lot of people, I mean, we struggle to get, you know, people who've got symptoms to get diagnosed with MND, and that's one of the problems, isn't it, that we wrestle with.
Yeah.
How would you possibly get people who've not shown symptoms involved in a drug trial?
Yeah, so this is for people who, so if MND runs in a family, well, first of all, I should say that just if you have a gene change, it doesn't necessarily mean to say that you're going to develop MND. That's really important to understand to start with. To answer your question, so if MND runs in a family and they've had genetic testing which has shown that the person with MND carries an SOD1 gene change, so carries a particular gene change, then they might decide with the right counselling and information, because there's lots of other things to consider, some genetic testing for other members of the family may say, yes, I want to be genetically tested. And so they would be tested if they wanted to be, and they might be shown to be carrying the SOD1 gene change. But those people have no MND symptoms. So that's the first thing. So if you can collect enough people, identify enough people who carry the SOD1 gene change, but they have no symptoms whatsoever, get them all together, and then all of those people, you do a blood test to measure their neurofilament light chain, which we think is a biomarker for MND. So if you have MND, that level of that neurofilament light chain level goes up. So you measure the neurofilament light chain in all of those people, and you do that over every month. Everybody gets measured neurofilament light chain. Now, any of those people in that pool, if their neurofilament light chain starts to rise over a 3-month period, that's an indication that they may or are most likely to start to develop symptoms of MND. And there's a time lag between the increase in neurofilament light chain and the motor neuron loss, because we think you need to lose lots and lots of motor neurons before disability happens. So as those people, their neurofilament light chain rises, then they are given the medicine. So the idea behind that is that they're not symptomatic and they're getting the treatment early on, and so they don't produce the toxic protein, the clumpy SOD1 protein. And so it's hopefully effective early on, if that makes sense.
Yes.
It's a revolutionary approach, and there are many different things to be overcome. You know, you want to be genetically tested, but it's people who generally would be anyway, because they you know, they're not forced into. Obviously, it's people who have that particular change. Does that make sense?
Yeah, thank you.
Yeah.
Yes. So, and anybody else? Any other questions?
Well, actually sort of on that, I presume that obviously a family that has a father or mother die, how would they know that they could put themselves forward for this?
So this would come through their neurologist, really. So neurologists in the UK are aware of this clinical trial, and so they would inform somebody that would be, they could possibly go on to that trial. So there's a group called the Clinical Studies Group, which myself and Brian are part of, which is the neurologists that meet every month to discuss what trials are ongoing, where they need people to recruit to, new companies that want to test, want to do clinical trials, etc., etc. So that would, just to go back a step, so if you had a family history of MND, then you can, if you wish, be genetically tested. In the UK at the moment, and this is something that we're all looking at and working towards and trying to understand whether it's a good thing or not, and how it will work. At the moment, only if you have a family history can you have a genetic test to see whether you have a gene that may or may not cause MND, with the appropriate genetic counselling as well, because there's lots of things. Do you want to know? You're not necessarily going to get the disease if you have the gene. You know, do you want to tell other family members? There's all kinds of different moral issues with that.
Yeah.
Thank you.
Do you feel more positive? I mean, I know it's a really difficult question, but do you feel more positive? You know, you show us all of this stuff. Do you feel more positive in the last couple of years, with the amount of money that's coming through now, maybe to research?
Yeah.
And some of the new things that you're showing us there.
Yeah, I think we definitely, yes. I think that we're at a kind of a revolutionary point now. So rather than it being pure guesswork, which it has been in the past, I mean, it's still very, very difficult, but there are some of these more targeted treatments, and they've been shown to work for some diseases. So if you'd asked me that question 2 years ago, I was extremely optimistic that this targeted therapy is going to be absolutely amazing and really excited about it. But there's been a bit of a reality check, as I mentioned, that SOD1 trial where the marker goes down, which is great, but the people, the symptoms in the people, it didn't show any difference. So that's a real disappointment, but it has got the positivity that the drug is reaching its target. So there's a different way to approach it. But that's really new technology, and there's more and more discoveries all the time and more information. So we're learning a lot about it. And there are examples where these gene therapies have worked. So there's a neurological disease called spinal muscular atrophy, and that affects children. And it's a protein that's missing called SMA. And there have been 2 different gene therapies for that. So in the clinical trial for that, 18 children were in that trial, and normally they'd all die within 2 years. And they were all given the therapy on compassionate grounds, and every single one of them survived and met their developmental milestones, etc., because this gene therapy was able to deliver the gene that was missing in those people. So there are some good examples of that, in addition to some cancers that have been, where cells have been treated outside the body back into people, etc. So yeah, there's a sort of shift in the way things are done, and certainly more and more awareness and more funding and more projects. Like, people actually want to work in MND. There's lots and lots of researchers who have moved from other diseases to work in MND because it's something that's tangible. There is a treatment out there somewhere, and a reason why people get it. And I think that's something that we know we're all optimistic that we'll find the answer at some point.
Thank you very much. Thank you.
Thank you. It all seems so positive. So many new things coming up. And thank you so much indeed for talking to us tonight. Now, are there anybody else that have any questions? No?
I don't think so, no. Just marvellous talk, thank you.
It's brilliant.
I think it's interesting that when people are on Riluzole, it's been out for such a long time now, and it's the only medicine, that did you say, Nick, that the exclusion criteria was going to change, and patients that have been on riluzole, for example, may be able to be take part in new trials, because that wasn't the case before?
Yeah, I didn't say that, but that's definitely true. So the Amylix trial and some of the magnet trials as well, people have been able to be on riluzole and take part in the trials. And what one of the things that's happening in criteria to enter trials now is that there's a lot of I guess it's computer based, so there's an understanding of where somebody would be and what their trajectory might be, depending on when they're diagnosed, when they first develop the symptoms, etc. So you can match two people who may have had MND for a longer amount of time, or very slow progressors, that would be able to get into trials, whereas previously there's a very, very tight entry criteria.
Yeah.
I mean, that's really important, and it feels like we're almost getting to the point where there might be too many trials. You can never have too many trials, but there'd be more trials than the people to take part, which would be, that would be fantastic. Or would it? I don't know. I can't think about which way around that is. But yeah, that's where we want to be. Lots and lots of effective, good trials that are all full. Because it's not, it's not right that we've got trials ongoing that people can't get into.
That's great. Thank you.
Yeah, thank you very much.
Thank you very much indeed. And thanks for coming along to talk to us tonight, Nick. And it's really reassuring to know it's all going in the right direction. And maybe we're nearly there. Let's just pray and hope.
So, yeah, thank you very much. Thanks. And
Thank you.
Any questions, please feel free to get in touch with the team. More than happy to talk about anything.
Thank you.
All right. Enjoy the rest of your evening.
Thank you.
Bye-bye.
See you again. Cheers. Bye. Okay. What's next?
I think we haven't got any long service awards, is that right? That I'm aware of, which is the next thing on the agenda.
Yeah, no, I spoke to Becky. It's all gone a bit pear-shaped with the long service awards, because I think Anne Coleman's been having one practically every year for the last 5 years. So I'm not sure what's going wrong up in head office with that. But for my records, no, there isn't anybody this year.
In that case.
Unless you think you have got one, then
No. I was just looking at the agenda, that was all.
Yeah. No, I don't think there are any.
Then, Barbara, were you going to talk about the Thumbs Up Club?
I am. Yes. So the Thumbs Up Club. For those of you that are unfamiliar to the Thumbs Up Club, let me enlighten you. It's fun for everyone and funds for the branch. Shortly, our treasurer, Alistair, will draw out this month's winners from the pot. You can subscribe to the Thumbs Up Club at £12 per subscription, and subscriptions aren't limited. You can have as many as you like. Each month there's a first prize and a second prize. First prize of £15, a second prize of £7 is drawn, and at Christmas there are 4 prizes. The first prize is £30 going to second prize at 20, third prize at 15, and fourth prize at £10. The prizes represent 50% of the subscriptions and are set for the year in January. However, you may subscribe to the Thumbs Up Club at any time at all. And if you would like to subscribe, please contact Alistair at any of our meetings, or email him on, shall I read it out, or have you got the email there, Alistair?
Treasurer Mnda. It's easier.
Yeah, it's mnda.wss.treasurer@chatham.plus.com. And okay, Alistair, who are we going to have this month? Who are the winners? Here's the bag.
Where's your beautiful assistant, Alistair?
I'm afraid my usual assistant is not up to it tonight. She had an operation last Friday. As I think. Yes.
well from us.
Yeah.
Yeah, so well.
She's feeling a little sore and a little tired, which is why she's not tonight.
Bless her.
Okay, number 1 for this month is number 58. And if I have another go. Sure, that's well rumbled. Number 2 is 44. So I will be in touch with the people who own these numbers tomorrow and advising them of their win.
Brilliant. Thanks, Alistair.
Well done.
Okay, has anybody got anything else that they'd like to bring up? No? Well, I think we can say that it's been an informative, interesting evening, and I'm so pleased that we had a very good turnout tonight. So I'm hoping the next meeting, which will be on the 27th of June, will be face-to-face, but we will see, and we'll let you know along the way. So I can wish everybody a good night.
Thank you. Thanks, Bob.
Thank you. Thank you very much.
Thank you very much.
Bye.
Bye.
Good night, everybody.
Bye-bye.
Good night. It was a great meeting. Nice to see everybody. Night, night.